All right, again, good afternoon. Good afternoon. While we're waiting for everybody to get in, missing a couple of folks, if you would pull up. Okay. Okay. So I take it when you log in on your phone, it's not going to show up in GoGuardian, I guess. But thank you for letting me know because I do attendance from GoGuardian. So I do need to know stuff like that. All righty. Missing two people. So okay, I'll go ahead and get started. So again, take a minute to the uh, vocabulary and introduction page in Canvas. The module is What is DNA? Or actually, it's structure and function of DNA lesson. That's what we're looking at. Okay. All righty. All right, so we're going to do, first we're going to look at that introductory module, the video on Canvas. So definitely log into Canvas, and then we're going to do a Nearpod um, activity with DNA and RNA. Okay. So actually just missing two people. All right, so let me do attendance real quick, and then I will share. So again, you should be logged into Canvas, the lesson on DNA structure. Now, we've kind of talked about it a little bit at the beginning of the semester when we were talking about macromolecules. So we talked about what DNA is made of and the fact that its units are nucleotides. So we're going to be looking at that more specifically this time. Okay, let me get this. Usually right after I just mark somebody absent, that's when they pop in. All right, but I appreciate y'all being on time. And so I can do the attendance and get that out the way. Okay. So I can share now. And just like I said, as soon as I do that. All right, let me get this person in. Let me mark down. Present. All righty, and now I will share. All righty. Oops, wait a second. Let me come back out again. Always forget to click that sound button because we are going to need that. Okay. Okie dokie dokie. All right, so we are in the module for DNA and protein synthesis. Actually, I didn't mean to click on that one. We want structure and function. So as always, the first page gives you a vocabulary, and you should always take a look at that. With this particular unit, there is a lot of vocabulary and a lot of technical vocabulary. See, this list is yay long. So take a few minutes to Like I said, some of this we've talked about before. We've talked about nucleotides. We've talked about the fact that DNA is double helix. So a little bit of that is going to be repeated. But as you can see, we're going to go much further with our DNA molecule than knowing what it's made of. We're talking about what it does and how it does it. All right, so just to do some historic background, we want to look at this timeline. Mine is taking its sweet time loading. There we go. All right, so on this timeline, I'm going to go through and play it, but as always, you can, whoops, go back and play it for yourself. When we do the recap, we're going to go ahead and do the Nearpod for the next two. So we're really just going to do this first part so that you get some historic background to DNA.
Miescher discovers nuclein. Friedrich Miescher was the first scientist to isolate nucleic acids from cells. He did this by collecting the white blood cells from the dressings of wounded soldiers. He found that this nuclein molecule, located in the nucleus, contained phosphorus, carbon, hydrogen, oxygen, and nitrogen. Nuclein was later named nucleic acid. Miescher and other colleagues at the time did not realize that nuclein was the molecule of heredity. Griffith mice and DNA transformation. Frederick Griffith conducted experiments with two related strains of bacteria. Rough coat bacteria did not cause pneumonia in mice. You can see this in the first column of the diagram. Smooth coat bacteria did cause pneumonia in mice, and the mice died. The reason for this is that the smooth coat actually protects the bacteria from the mouse's immune system. Interestingly, Griffith took this experiment 2 steps further. He then decided that he would try to kill the smooth strain of bacteria first before injecting it into mice. He did this in the third column of the picture. The mouse lived. Finally, Griffith decided to mix both the killed smooth strain and the rough strain together and culture them for a few days before injecting them into the mice. When he did this, he found that the mice died. The conclusion that Griffith made was that transformation had occurred. Some unknown chemical in the dead cells was transforming the harmless rough cells into dangerous smooth cells. We now know that this chemical was the DNA from the heat-killed cells that was not destroyed. Bacteria can actually take up the DNA from their environment and use it to make smooth coats, making them more deadly. Avery, MacLeod, and McCarty. These scientists purified and tested all the chemical components in the smooth and the rough bacteria from Griffith's experiment to determine which component caused the transformation. Their conclusion was that this was likely DNA, but at the time very little was known about the composition or the functions of DNA. In fact, most scientists still thought that proteins were the hereditary or the instructional material for cells. Chargaff's rules. Chargaff studied the molecule of DNA and determined that there was always the same amount of the chemical adenine as there was thymine, as well as the same amount of cytosine as there was guanine within the DNA molecule. His conclusion was that adenine and thymine must form pairs, and then the same would be true of cytosine and guanine. Hershey, Chase, and blenders. Hershey and Chase experimented to find out if the transforming principle from Griffith's experiments was DNA or if it was protein. Hershey and Chase used a virus called a bacteriophage that infects bacteria. They knew that bacteriophages were made of 2 things. One was DNA, which contained phosphate, and the second was protein, which has sulfur. Which molecule would enter the cell to make more viruses? They labeled the phosphorus and sulfur, each with radioactivity, and observed what happened in 2 experiments. Look at the diagram. Since the protein coat of viruses contains sulfur, the viruses were treated with radioactive sulfur that would bind to the protein but not DNA. and be easily detected with special equipment. This is shown on the left-hand side of the diagram. On the right-hand side, you can see that since DNA has phosphate molecules and not sulfur, the viruses were also treated with radioactive phosphate that would bind to the DNA but not the protein. The treated viruses both infected the cells, and then the cell parts were broken apart in, of all things, a blender. They separated the pieces into groups and determined that the DNA with phosphate was the material that was being injected into cells, and not the sulfur-containing protein. Radioactive sulfur was found on the surface of bacterial cells, and radioactive phosphate was found inside the bacterial cells. The conclusion here was that only the genetic material, DNA, entered the cells and caused transformations.
All right, and last but not
In
least.
1953, the structure of the DNA molecule was revealed. James Watson, Francis Crick, and Maurice Wilkins examined an amazing photograph made by Rosalind Franklin, a colleague of Wilkins, using a technique that she developed called X-ray crystallography. Together with this information and the studies of many other scientists, Watson and Crick deduced a model for the structure of DNA. For this, they all received the Nobel Prize. Their model for the DNA molecule consisted of two nucleotide chains that wrap around each other to form a double spiral. This shape was called a double helix.
Okay. With Rosalind Franklin, just a trivial fact, they took her information, but at first they didn't give her credit. So it wasn't until after she died that she got credited and a Nobel Prize for having come up with the original blueprints for DNA. But when we study DNA, everybody knows Watson and Crick as the founders. In fact, as we watch the Nearpod activity, they're going to start with Watson and Crick. Okay. All right. So this is just a little historic background on some research that they did to basically come up with the fact that the genetic material is DNA and it is double helix. So when you get that question with the Nearpod, what do you know about DNA already? That's what you know. All right. So I'm going to give you the code in just a second. And we're going to do this together. So and while we're waiting for this, make sure that you've done your DNA Halloween story. A lot of you have not done that yet. So make sure you've turned that in. And it needs to be scientifically based. All right. There we go. Copy link. Copy the link. There we go. All right, so the link is in the chat.
This is DNA. This is the molecule we use to store our genetic information. DNA contains the instructions needed for an organism to develop, survive, and reproduce. If you want to understand how DNA works, the first step is to learn about its structure. The structure of a molecule determines its function, so making sense of the structure is essential for you to understand how DNA can do its job.
Okay, so I'm going to pause this, put this on student view. All right, so one of the first things she said was we need to understand structure in order to understand function. So that's basically what we're going to be looking at. What is the structure and how does this structure function to do the job of transmitting genetic material? That is our goal. So give you a few minutes, make sure it didn't look like everybody was logged in yet. So go ahead, get logged in. Answer that warm-up question. All right, again, you're reminded that I do at the end look back at the transcript, and you get your participation grade by what the report tells me at the end. Did you answer all the questions? Did you do the polls that they asked you? If there's a quiz, did you do the quiz? What score did you get on the quiz? So all of that is recorded in the Nearpod. Whether you participate or not determines what score you get at the end. So please participate. LaCoria, are you able to do this on your phone? Okay, good. All righty, so let's take a look. All right, we still got a couple of no answers. All right, let's go, folks, because once I move on, I'm moving on. Thank you. All right, so it is the instruction on how to make the body or any to code for code for a video game. Well, I guess they kind of they would call it if you're doing a video game or blueprints for the house, because it is the blueprint for living things. So we're not going to worry about video games and houses, but we are going to look at for making living things. That's where our concern is, okay? Y'all let me just give you a heads up. When y'all go and look up Google an answer, make sure you Google an answer that suits the class. Don't just copy whatever it says, because sometimes they give you a definition that goes with something else and not necessarily a biological answer. All right, it's like we're not gonna look at codes for gaming codes or house blueprints. It is a double helix. This is true. A lot of data is stored in DNA. Okay, Kayla, we still have no answer. Let's go. What you doing? All right, but I'm moving on. Please stay
In
watching
April
what
of
she's saying.
1953, James Watson and Francis Crick changed the world of biology forever with a landmark paper published in the journal Nature. It was just one page long. They reported they had determined the structure of DNA was an anti-parallel double helix. How did Watson and Crick figure this out? They didn't do any experiments. Instead, they built models out of tin and wire that matched measurements derived from other scientists, including biochemical data from Erwin Chargaff and X-ray diffraction data obtained by Maurice Wilkins, Rosalind Franklin, and Raymond Gosling under the direction of John Randall. You may have tried making your own DNA molecule using a chemistry molecular modeling kit. Today, we're building models of DNA using a 3D printing pen. MorphPen was kind enough to send a 3D printing pen to Socratica for us to test out. So of course, we're making DNA with it, obviously. You can make whatever you want with your 3D pen. Single-stranded DNA is a polymer of nucleotides, made up of repeating units of sugar, phosphate, and nitrogenous base.
All right, so remember way back, we had our big macromolecule worksheet. We talked about monomers and polymers and what the individuals were made of. Okay, so you might want to find that worksheet. Okay. I shouldn't see 25% no answer. Everybody needs to have an answer, folks. All right. But as we can see right now, all of the above is correct because a nucleotide, if you remember, is made up of a nitrogenous base, a phosphate unit, and a sugar. All of those things make up one unit, and then they're connected together.
In double-stranded DNA, two polynucleotides are attached at their complementary bases, zipping the two strands together with hydrogen bonds. Keep in mind, most DNA molecules are very, very long, thousands or even millions of base pairs long. We're just going to make a very short stretch of double-stranded DNA as an illustration. DNA looks like a twisted ladder. The sides of the ladder are made of sugars and phosphate groups. These repeat over and over: sugar, phosphate, sugar, phosphate. This is the backbone of DNA.
Okay, go ahead and answer. Which molecules are the backbone? So we got three things. Not all three of them are the backbone. All right, I find that this was a kind of a hard question to answer because two of them, they don't give you the opportunity to answer two of them, but sugar and phosphate, they are what we call the side, what we call it, we refer to DNA as a ladder. And so the sides of the ladder are made of the sugar and phosphate. So they're actually only giving you one way to answer this question, but both answers are correct. Sugar and phosphate together make up the rungs of the ladder.
Let's first make a backbone using our 3D pen. We're actually going to need two of these, one for each side of the ladder. Now, let's look at the rungs of the ladder, which is really the business end of DNA. These rungs of the DNA ladder are made up of 4 different kinds of nitrogenous bases: guanine, adenine, thymine, and cytosine, abbreviated GATC. The order of these bases is how information is stored in DNA. Think of it like the order of letters in a word, and the order of words in a sentence. Guanine always pairs with cytosine. Adenine always pairs with thymine. These are known as Chargaff's rules, in honor of Erwin Chargaff, who determined this biochemically.
Okay, so take a minute to answer that question. And again, this was something that we had talked about earlier in the year. So again, she referred to this as the runs of the ladder. That means that the steps, when you're going up a ladder, the steps are called the runs of the ladder. But imagine that it kind of zips down the middle. So you had your backbone on one side and then a nitrogen base. And then you have a backbone on the other side and then a nitrogen base. So those two nitrogen bases meet in the middle. And I think she said earlier that they are held together by hydrogen bonds. Okay. So, and I said, imagine it zipped. Later on, we're going to use that phrase. It's going to unzip. Okay. So we have, imagine that we have this ladder. It has two sides and then it has runs, but imagine that it actually has a cut down the middle of it. Now, Chargaff's rules are complementary base pairs. So who goes with who? That's what it's asking you. Which pairs always match with each other? And this is going to be important because you're going to have to decode a DNA. So you're going to have to know which pairs match all the time. Okay, got a lot of no answers, folks. Let's go. Let's go. Stop waiting for it to pop up. Just copy somebody else's answer. Okay. Thank you, Kendall. The rule says A, and it's not so much that they equal, that they bond with. Okay, they bond with. A always bonds with T and G always bonds with C because we're talking about bonding or connecting. Okay. Okay, and because they're the ones that bond together, like Victoria said, there will always be an equal amount of guanine and cytosine, and there will always be an equal amount of adenine and thymine because they bond, they pair, they're base pairs. Okay. All right, again, folks, I'm not sitting back waiting for you to think about answering. You're supposed to be keeping up. They basically give you the answer and then they ask you the answer. Just you're listening.
Notice that there are 3 hydrogen bonds holding G and C together, while there are 2 hydrogen bonds holding A and T together. Let's take a closer look at these components. The 5-carbon sugar in DNA is deoxyribose. Deoxy meaning it's missing an oxygen compared to regular ribose. The phosphate group looks like this. This is where it's attached to deoxyribose. The phosphate groups are acidic. Think of them like little acidic flags hanging off the structure. This is what gives DNA its acidic nature when in solution in water. Now, how about those nitrogenous bases? They're called that because they're literally basic as opposed to acidic, and they all contain nitrogen. 2 of them, adenine and guanine, look like this with 2 fused rings. These bases are called purines. The other 2, cytosine and thymine, look like this with only 1 ring. These bases are called pyrimidines.
All right, I'm going to pause it there because they're about to ask you that. So I'm going to give you the opportunity to write that down. Purines have double rings, and pyrimidines have single rings. And you need to say purines are adenine and guanine, and pyrimidines are cytosine and thymine. Purines are double rings, pyrimidines are single rings. They're about to ask you that question. All right, so which ones are pyrimidines? Which you just wrote down. And again, I guess this is kind of hard to do because it should be, because there's 2 choices. Okay, so for those who answered the question, yes, our 2 answers that are pyrimidines would be our cytosine and the thymine. And the guanine and adenine
Since adenine always pairs with thymine and guanine always pairs with cytosine, you can also say that a purine always pairs with a pyrimidine. One unit or monomer of the DNA polymer is called a nucleotide. This is one sugar, one phosphate, and one nitrogenous base. In the case of DNA, they're also called deoxyribonucleotides, as opposed to in RNA where they're called ribonucleotides because they contain ribose instead of deoxyribose. Nucleotides are joined together by covalent bonds. The phosphate group of one nucleotide is linked to the hydroxyl group on the sugar of the next nucleotide. These are known as phosphodiester linkages. Here's a technical note. Biochemists have a particular way of labeling the atoms in DNA so they know what part of the structure they're referring to. This is an important shorthand. So you can just say something's happening at C5, for example, rather than having to point it out on a diagram. They start with labeling the skeleton, the atoms in the ring area, of the nitrogenous base. You can see here these atoms labeled 1 through 6, or over here labeled 1 through 9. Then, to label the skeleton of the sugar, they don't want to use the same numbers that they used for the base because that would be confusing, so they label them 1', 2', 3', 4', and 5'. The 5' carbon is this one that sticks up from the ring. This is where the notation 5' to 3' comes from. You may have heard that phrase, DNA runs in the 5' to 3' direction. That's talking about one of the two strands. You can see the sides of the ladder run in opposite directions. Remember, it's an anti-parallel double helix. On one end of a DNA strand, there's a phosphate group attached to the 5' carbon, and the other end of that strand has an OH group on the 3' carbon. We say this side runs in the 5' to 3' direction because of the labeling of the atoms. The other side of the ladder runs in the 3' to 5' direction. This is going to be important to remember later when we study how information is stored in DNA. One side is the coding strand or sense strand, the 5' to 3' side. The other side is called the anti-coding strand or antisense strand. Because of complementary pairing, if we know the sequence of one strand, we know the sequence of the other strand. It's kind of like a photograph and the photographic negative.
All right, we have a question to answer. Right, so put it in your own words. Try not to go Google it and copy and paste an answer. It says in your own words. And it's very simple to say. You don't even have to say a lot. Anti-parallel means something very simple. Okay, I got one answer. Jacob says that the end of the 5 strand is paired with the 3 end of the complementary strand. That is true, but that's really not explaining what we mean by anti-parallel. Folks, all this anti-parallel, anti-anything means that it goes against. So when you think of 2 strand, thank you, Kayla. 2 strands run in opposite direction. That's all that means. We have our double helix, and one strand is going in one direction and the other strand is going in the opposite direction. They are parallel to each other, but they are not exactly the same in that they are traveling in different directions. Okay? 2 strands running in opposite directions and allow for complementary pairing. That's good too. Okay. All righty. One more second for those of you who did not answer to answer. Something. Thank you. But Corey, I have you in here twice. Why is that? All right, moving on.
Watson and Crick, rather cheekily, said at the end of their paper that the structure of DNA suggested a mechanism of replication. And they were right. But we'll talk about DNA replication another day. That's a very long, complicated, and serious story. Back to the DNA structure. Remember we said Watson and Crick were guided by X-ray diffraction data. That's how they knew the spacing of the components of DNA. Their physical model had to match the values derived from the X-ray photographs from Gosling, Franklin, and Wilkins. This is what they determined. The radius of DNA is 1 nanometer. This is what you get if G pairs with C and A pairs with T. So this also satisfies Chargaff's rules. If you don't pair G with C and A with T, you get a strand that's either too fat or too skinny to match the X-ray diffraction data. The two polynucleotide strands wrap around a central axis to form a right-handed helix. The helix makes one full turn every 3.4 nanometers, and there's a 0.34 nanometer distance between nitrogenous bases. This means there are 10 layers of base pairs, or rungs, per turn of the helix.
Okay. All right, and that's real technical information, but what did she say? In other words, there's a pattern to the way that it twists, and it's going to keep repeating the same twist every so many base pairs. Okay. All right, most of you are getting it. It's every 10 base pairs, it's going to repeat and make that ladder twist.
Notice there are two different kinds of grooves made as a result of the twisting of the helix. In one groove, the backbones are far apart, and that's known as the major groove. Then we see here where the backbones are close together, and that's called the minor groove. This plays a significant functional role because there are specific DNA binding proteins that will only bind at certain locations, and some of them bind at the minor groove and others bind only at the major groove. Now that you know all about the structure of DNA, you'll be prepared to learn about its function. I have to tell you, this is a huge subject. Really, there's an entire field of biology devoted to the function of DNA and other biological molecules, called molecular biology. I personally am a molecular biologist, so I plan on guiding you through that study.
Okay. All right, so take a minute to answer that question and then we'll move on. I don't know, some of you say, I don't care about DNA one way or the other, but you should. This is you. You are made up of, everything about you is controlled by the DNA. So it should make you curious as to how you do what you do.
So, subscribe so we can let you know when we release each video. If you want to make sure we keep making these gorgeous, really stunningly beautiful science videos, there are a few ways you can help. Make sure you watch to the end of a video.
Okay, we are not going to worry about the end of that, but we are going to go back to Canvas for a minute. All right, so basically, oh, I'm sorry. Let me see if I can get back there. Hold on, hold on. Yep, there it is. Well, I didn't log out of it, so you should still be able. Can you not answer it because I went on? Okay, there we go. That's an interesting. Okay. All right, so we'll take it that only 3 people. What role? You still writing, Jacob? All right, so I'm going to take it that the rest of you are not curious at all, but you should be. All right, so we are going to look at DNA structure. We are going to talk about, you are going to have activities where you have to match up DNA, especially when we do protein synthesis. You're going to have to be able to create a protein by knowing base pair sequencing. So it's going to be important. All right, so I'm going to come out of this and come back to, see what time is it? Come back to Canvas. Okay, so I'm not, you can watch this one. This one is very short and it kind of goes through what she just did in the video. And let's just take a minute to go through these turn cards. All right. Okay, so this is the type of thing that you're going to have to do. They will always be giving you base pairs, a base pair sequence, and then asking you, what is it going to look like on the other side? So if they give you A, T, G, G, C, A, your job is to remember that every time you see an A, it's going to pair up with a T. And every time you see a T, it's going to pair up with an A. So write that sequence down right there, A, T, G, G, C, A. And then underneath it, so don't write it behind it, write it like they are pairing, right? T, A, C, C, G, T, because that's how it's going to pair. Okay? And like I said, you need to make sure that you understand that G is always going to match with C and A is always going to match with T. For DNA, when we look at RNA, we're going to have one slight change, but basically understanding base pairing, you will get chains. So your test questions will always say, if this is the sequence on one side, what's the other side going to look like? And you need to be able to match up the other side in correct sequence. Okay? All right, let's take a look at RNA. All right, so basically you're going to have to know the differences between the two. Okay. All righty. So just, I do have a Nearpod for this, but I'm going to let you just get the information off of Canvas for this one. So looking at, if we're looking at they have them side by side, DNA and RNA. One of the things that she talked about is RNA. DNA is made up of a sugar called deoxyribose, and it's called deoxyribose because it's missing an oxygen. That's for the deoxy part. Okay, so one of the differences is the type of sugar that it includes. So DNA has deoxyribose, RNA has ribose. That should be easy to remember for the starting letter, D and R. Okay, that's the sugar that they're made of. Secondly, they have the same base pairing except for one. Okay, if you notice this, so you have cytosine, guanine, adenine on your RNA, but then you have this one called uracil. So that one is only found on RNA. Okay. What's going to happen is it replaced the thymine. So there is no thymine in RNA. Okay. But A is supposed to bind with T. So now uracil on an RNA will bind with T. Okay. So it's still going to be the same base pair, except it's going to be it's a different nitrogenous base. But when we get to pairing, it will still pair. Everywhere there was everywhere there was a T, this will bond with A. T and A bond together. So the uracil will bond with adenine because A and T bond together. So your guanine is still going to bond with cytosine. But instead of A and T, you're going to have A and U for RNA. Okay? All right, so you have this rule down here. So that's a difference. So you have to know what the similarities and differences are. That's always a test question is all. Compare and contrast the structure of RNA and DNA. All right, then you need to know that there are 3 types of RNA. And this is important because as when we start talking about protein synthesis, as to what each one does. They all don't do the same thing, even though that they're made up of the same thing. Oh, I forgot the other one is RNA is single-stranded. DNA is double-stranded. One other very important thing is DNA never leaves the nucleus. It cannot get out of the nucleus. So it needs the first one. It needs a messenger RNA. So the messenger RNA's job is to copy the message, and then it can leave the nucleus and go to the ribosome. So again, earlier we studied what do all the parts of the cell do? The ribosomes make protein. So that's going to be our major destination. When we leave the nucleus, we're headed to the ribosomes because that's where we're going to make the protein. So the job of the DNA is to code for protein synthesis. Okay, so it has the blueprints, it has the instructions on what to do, except it stays in its office. Like think of the nucleus as the main office. And it has a little foreman come in. He gets all the information from the office and he takes it down to the workers who are actually going to make the proteins. Okay, so that's how you have to think of RNA and DNA. All right, so again, you have a messenger RNA. It's going to actually make a copy of the DNA. Then you're going to have a ribosomal RNA, which is actually located in the ribosomes. And it kind of sets itself up as a template. And we're going to look at how it does that. It sets up like a template. And then the transfer RNA is going to say, oh, you want me to bring this part to you so I can make the protein? So that's what the transfer RNA goes and gets the part, in this case, the amino acid, and it brings it to the ribosomal RNA, and then they connect all the pieces together to make the protein. Okay, so each of them is located in a different place, doing a different job. All right, all right. I'm going to let you take a few minutes. I'm looking at the time. Let's just spend the rest of the time working on this, putting this one together. So take a few minutes to do that, and then we'll look at what the correct answers are. And we'll do the RNA Nearpod on Wednesday. So again, you're going to drag the correct answer. What's the difference between DNA and RNA? And I see two people trying to do it. Okay. So when I'm looking at GoGuardian, I should see everybody doing the drag and click. And I don't see everybody doing that. Everybody should be doing the drag, pull and drag for this is practice. Okay, maybe we'll do this as a little quiz on Wednesday. Okay, go. Hopefully you've checked your answers. Those of you who have done it, and I guess some of y'all are waiting for us to do it for you. All right, DNA we said is double-stranded. RNA is single-stranded. The D in DNA stands for deoxyribose, and that is the sugar that it's made of. Remember, our nucleotide is a sugar, a phosphate, and a nucleotide. I mean, a nitrogen base. So the sugar is here. Uracil is only found in RNA. There are 3 types of RNA. Thymine is the base that's only found in DNA, and DNA is only found in the nucleus. Okay? All right, so those are the things you need to know. So like I said, I'm going to put together a little quiz for Wednesday because some of y'all weren't participating. So between now and Wednesday, make sure that you've read this page and you are familiar with this information. All right, we got a few minutes left and a couple of you still owe me some things. So we're going to use those few minutes to turn in any, because folks, the other things, the due date is due, and I told you I'm going to let it give you a 0. The grade book automatically puts a 0 in when the due date is reached. So if you have something outstanding, and most of you have your Halloween story is missing. Most of you. So take a few minutes to get that turned in because that's going to be late. And progress reports are going out next week. So you want to make sure all your missing assignments are done.